To understand the Krebs cycle, we'll be looking at our friend here who's eating a sandwich. After this is digested, it will be entering the Krebs cycle. So the Krebs cycle starts with the molecule acetyl-CoA, which is hydrolyzed to form a molecule called citrate. From here, citrate is oxidized, and a electron is going to be transferred from NAD plus to form NADH, an electron transporter that can then be used later on in the electron transport chain. But from here, we have a loss of a carbon molecule in the form of carbon dioxide, and so now the 6-carbon molecule of citrate becomes a 5-carbon molecule of alpha-ketoglutarate. Alpha-ketoglutarate is then oxidized, and again, in this transfer of electrons from NAD plus to NADH and the loss of another carbon will form a 4-carbon molecule which combines with coenzyme A, or CoA, to form succinyl-CoA. From here, this forms another molecule that has a phosphate instead of the CoA because it's pretty unstable, and that phosphate can then be combined with GDP to form GTP, which is like energy, like ATP for the cell. From here, we have a 4-carbon molecule called succinate, and after another oxidation reaction and a transfer of electrons from FAD to FADH2, we'll end up with a carbon molecule called fumarate. Fumarate is then going to gain a water molecule and form a compound called malate, which again, still 4 carbons. And then entering that last transfer of electrons from NADH to NAD plus to NADH, in that final oxidation, we'll have oxaloacetate. So this carbon molecule is going to form enough carbons for this to go around the cycle twice. So there's 4 of them. We can see there's 2 in the acetyl-CoA molecule. So this is enough to regenerate that cycle and take it around for the second time.